Once I rose above the noise and confusion, just to get a glimpse beyond this illusion. I was soaring ever higher, but I flew too high, masquerading as a man with a reason. My charade is the event of the season, and if I claim to be a wise man, it surely means I don't know. Carry on, my wayward son. There'll be peace when you are done. Lay your weary head to rest. Don't you cry no more. Carry on.
Hello and welcome to Bio40 Pre-Lecture 33. Today we're going to discuss designer babies. The term designer babies refers to children whose traits have been determined based on genetic screening or gene modification. There are three main ways the traits of a child can be controlled: selective abortion following genetic screening, pre-implantation genetic diagnosis before in vitro fertilization, and embryonic gene editing. The last one is mostly just theoretical at this point, but the first two are already being used. While not normally thought of in these terms, selective abortion is a first way that parents exercise control over the genetics of their children. Since the 1960s, it's been possible to perform genetic analysis of fetal cells extracted through the process of amniocentesis. Back then, karyotypes could reveal information like the sex of the fetus and chromosomal abnormalities such as aneuploidy. Today's technology has made early genetic analysis even easier. New methods for obtaining fetal DNA are less invasive and can be done earlier in the pregnancy. Cell-free fetal DNA tests only require a sample of the mother's blood and can be done within the first 10 weeks of the pregnancy. And genetic analysis has advanced such that the fetus's entire genome sequence can be known. Prenatal genetic analysis offers the parents the option of terminating a pregnancy if the fetus has an unwanted genotype. There is much discussion about sex-selective abortion as a possible explanation for the unbalanced sex ratio at birth that exists in various parts of the world. There's also selective abortion related to disease and disability. Frequently this means conditions like Tay-Sachs, where any child born with the disease will not survive to adulthood. However, the disability rights community is concerned with how the practice is used for conditions that are not lethal in childhood. A 2015 study suggested the number of children born with Down syndrome in the United States has been reduced by 30% due to abortions following prenatal genetic screening. It's important to consider the ethical implications of our technology because the science alone cannot tell us if an application is right or wrong. But this isn't really what we normally imagine when we talk about designer babies. The phrase evokes the idea of shopping for a baby's traits the same way we shop for goods in a store. So just how realistic is this idea? To some degree, choices like this can be made using pre-implantation genetic diagnosis prior to in vitro fertilization.
Your extracted eggs, Marie, have been fertilized with Antonio's sperm.
In vitro fertilization, or IVF, involves extracting the woman's eggs and fertilizing them outside the body. The zygote is incubated for several days and develops into an embryo. The embryo is then transferred to the mother's or a surrogate's uterus, where hopefully implantation will occur, followed by normal fetal development. But IVF isn't done with a single egg. That's because not all eggs are fertilized, not all fertilized eggs develop properly, and not all embryos successfully implant. The overall success rate of IVF barely exceeds 40%, even under optimal circumstances. So if a large number of eggs are fertilized and develop into embryos, the parents can have options about which ones they want to attempt to implant. and preimplantation genetic diagnosis, or PGD, allows them to know details about the embryo's genes that they can take into account when making the decision. In PGD, 1 or 2 cells are biopsied from the developing embryo. Thanks to PCR technology, this is all that's required to get sufficient DNA to detect polymorphisms and determine which alleles the embryo has. Detecting large-scale chromosomal abnormalities is a little different. It can be done with a procedure called fluorescent in situ hybridization, or FISH. This involves attaching fluorescent dyes to polynucleotide probes that base pair with specific DNA sequences of interest. Here, each chromosome is labeled with a different color dye in what's called spectral karyotyping, which can reveal the sex and detect aneuploidy and translocations. A given chromosome can also have different regions labeled in different colors, creating a specific barcode. This helps us see large deletions, duplications, and inversions. Once this genetic information is known, the parents have several decisions to make.
Naturally, no critical predispositions to any of the major inheritable diseases.
It's common to screen out embryos with genetic diseases, though other criteria are more subjective.
First, we may as well decide on gender.
We would want Vincent to have a brother.
Parents can decide to implant only XX or only XY embryos, depending on if they want a daughter or a son.
You have specified hazel eyes, dark hair, and fair skin.
Now this is what we imagine when we talk about designer babies. And these decisions are possible. Suppose one parent has blue eyes, which is homozygous recessive, and the other parent is heterozygous and has brown eyes. Half the embryos will be heterozygous and half will be homozygous recessive. The parent can choose to implant only the half that matches their preference.
I have taken the liberty of eradicating any potentially prejudicial conditions, premature baldness, myopia, alcoholism and addictive susceptibility, propensity for violence, obesity.
So here's where we get into the real dream/nightmare of designer babies. Exactly how much control can parents exert over every aspect of their child's appearance and personality? Well, let's imagine we want to find an embryo that has every trait that doctor discussed with the parents. How likely is this? We'll say some of those traits require a homozygous recessive genotype, while others can be homozygous dominant or heterozygous. Now suppose the parents are heterozygous for each trait, except sex of course. So we can calculate the probability of having the right genotype for each trait. Multiply them together, and we see the total odds of an embryo having the perfect genotype are 0.02%.
You could conceive naturally 1,000 times and never get such a result.
Well, yeah, but also good luck screening 5,000 plus embryos for the perfect one. And this was a major simplification. Most of these traits are really controlled by multiple genes. Even eye color actually involves 2 genes. Also, this isn't even considering traits like intelligence, creativity, athleticism, kindness, and so on, which are even more complicated. It might be necessary to screen trillions of embryos to find the exact genotype the parents want. For comparison, in IVF, doctors normally try to extract 10 to 20 eggs from the mother. And these calculations assume the parents at least had the proper alleles. Suppose the parents were both homozygous dominant for a certain gene, and they wanted their child to be homozygous recessive. I guess we could screen for the extremely uncommon embryo that happens to be the result of a sperm with a spontaneous mutation fertilizing an egg with a spontaneous mutation. But wouldn't it be better if we could make the mutations in a directed manner? Welcome to the world of gene editing. Genetic modification through selective breeding has existed since the dawn of agriculture. But the ability to edit a specific gene in a specific manner is a recent development, new to the age of molecular biology. We can take genes from one species and put them in another. Human genes can be expressed in bacteria. Here's a mouse that glows green because it's expressing a gene from a jellyfish. And we're not even limited to genes that naturally occur. It's easy to make DNA with any sequence you can conceive of. The question is, how do we get that DNA into the embryo's genome? Getting the DNA into the cell isn't an issue. But if it's going to be maintained, that DNA must become part of the chromosome. Also, when we add the desired allele, we normally want to get rid of the allele that was there in the first place. This could most easily be accomplished with homologous recombination. The desired allele would become part of the chromosome, and the undesired allele would be degraded. However, spontaneous homologous recombination is uncommon. How can we make it more likely to occur? Well, homologous recombination is normally used to repair double-strand breaks. So if we had a way to make a targeted double-strand break in the undesired allele, the repair system would use our desired sequence as a template, thereby incorporating the allele we want into the chromosome. Whatever we use to make the double-strand break would need to be very specific, though. It can't cut anywhere else in the entire genome, and it can't cut the very similar sequence we want to replace the allele with. We've already talked about restriction enzymes that catalyze double-strand breaks. The problem with them is that the sequences they recognize are typically only 4 to 8 base pairs in length. That's small enough that any given site would occur many times throughout a genome as large as ours. For a site to be unique in a genome of 3 billion bases, it probably has to be at least 16 bases long. There are proteins that recognize sequences that long, including zinc finger nucleases and homing endonucleases, which Pomona's own Professor Len Seligman researches. Much work is being done to figure out how to engineer these proteins to cut any desired site. However, the hot new technology for this sort of work is CRISPR-Cas9. This was actually discovered as a form of prokaryotic adaptive immune system. When a phage attempts but fails to infect some species of bacterium, part of the phage DNA gets saved in the bacterial chromosome. These regions are transcribed and serve as guides for the nuclease Cas9. If the same type of phage attempts to infect the cell again, the guide RNA will direct Cas9 to cleave the phage genome. We can take that system and replace the CRISPR sequences with one that will guide Cas9 to catalyze a double-strand break specifically in our undesired allele. Researchers like Pomona alumna Jennifer Doudna have done a lot of work in developing this technology. To date, it's been successfully used to alter the genomes of everything from plants to human cells and culture. Recently, a group in China made big news with the first attempt to use CRISPR-Cas9 in human embryos. To avoid ethical issues, they used non-viable tripronuclear, or 3PN, zygotes, which form when 2 sperm fertilize the same egg in IVF treatment. The CRISPR-Cas9 system was very successful at cutting the allele, but repair with the desired sequence was inefficient.
You're engineered just like the rest of us?
Not quite like the rest of you. An acceptable likelihood of heart failure.
Unfortunately, my son's not all that they promised.
But when it comes to engineering children, the technique simply failing to work should be the least of our concerns. The thing to really worry about are side effects that could severely impact development and potentially lead to major physical and cognitive impairments. I'm not sure that we could ever reduce side effects to levels we consider acceptable given the seriousness of the consequences. But why don't we just imagine that we eventually overcome these problems and reach a future where parents could control every single base of their child's genome with perfect accuracy? That still will not grant total control over the child's traits. First of all, many traits are influenced by the environment, which includes everything from the conditions of the womb to the schools the kid attends. Second, some traits are the result of stochastic processes. They're random. I think freckles are a good example of both of these issues. A child could be engineered with a predisposition for freckles, but it takes environmental influence, UV damage, for them to actually appear, and the pattern will be unpredictable. With more complicated personality traits, like intelligence and creativity, it's even more difficult to understand the interactions between genes, environmental factors, and randomness. Even the genetic portion alone is hard to fully know. These traits are influenced through complex interactions of many, many genes. Right now, the best we have are loose correlations between personality traits and alleles of certain genes. Even traits we think of as simple cannot be predicted with 100% accuracy. Scientists will continue to study these relationships, but it is a very real possibility that we'll never possess a total understanding of how genes influence our traits. So to summarize, parents can already exercise some limited control over the traits of their children. Features like the sex of the child and simple traits that are controlled by 1 or 2 genes can be screened for during the process of in vitro fertilization. Further manipulation through genetic engineering is possible in theory, But serious ethical concerns will stop this from being available anytime soon. And even with this technology, control of traits will always be limited because genes are only a portion of who we are. Which brings us to the final Bio40 life lesson. DNA is not destiny.